Look what we got here.
What happened, Detective?
A hit and run. Somebody stole money from the local gas station, lost control as he was escaping.
Look what I found, Detective.
Yes, he must have been hurt from the crash, trailed some blood.
This means we can take it back to the lab and analyze it with other DNA samples we found at the crime scene. We are going through the process of gel electrophoresis.
Yes, and can you remind me how to do that?
Sure, but first we have to get back to the lab.
DNA is deoxyribonucleic acid. The nucleotides are made up of a 5-carbon sugar deoxyribose, a phosphate group, and a nitrogen base. The DNA molecule is shaped like a ladder, called a double helix. In a DNA molecule, adenine always bonds with thymine, and cytosine always bonds with guanine. The purine bases, which are made up of adenine and guanine, have a double ring structure. There are also primide bases, which are made of cytosine and thymine, and have a single ring structure.
First, we have to introduce a restriction enzyme to the collected DNA samples to cut into pieces, and then dyed so it's easier to see in the gel. We have already prepared the suspect's DNA for this test.
Oh yeah, now I remember. We need to cast a special gel that as well as to hold the cut DNA, and the gel is porous so it allows the DNA to move through it when we run the electricity.
Next, the wells are filled with cut DNA of the evidence and the DNA of the suspects and is placed in the gel electrophoresis machine. And we will also fill the machine with a liquid called a TAE buffer, and electricity will be led from one end of the machine to the other.
Because DNA is negatively charged, it will move to the positive end of the machine, and because DNA is cut into pieces, the shorter pieces will travel faster than the larger ones, and this will create a barcode-like pattern.
Yes, now that we have the DNA spread out, whoever's DNA matches the evidence is the culprit.